Okay, so now you can see more windows, right? Okay, so when we talk about a frame, what we just mean, since this is a point scanning laser or imaging program, basically the laser is going to go through this image and scan across this whole line, okay, pixel by pixel, okay, all the way through one rectangle, okay, and that is called a frame, one rectangle, okay, and you can basically average a bunch of frames together, have it repeat this process a bunch of times and average them together, okay? So for example, if I say I want it to do one frame, I'm gonna grab an image of one frame, okay? It just did one scan. Oh, all right, hold on. Let me change this. Okay, it did one scan of whatever image, okay? Now let's say I want to, one thing you'll notice is that the image is kind of noisy, right? You can see it looks kind of pixelated. You've got some photons in the background. You can even see over here my FLIM curve looks kind of noisy. So one easy way to increase your signal to noise is to have it do this instead of just once, have it do it several times and average them, okay? So if I say instead I want to do 12 frames and I want to average them, okay, and I hit grab again, it's basically scanning that one picture 12 times and it's averaging them, okay? What this is effectively doing is it's increasing my real signal and getting rid of my noise. You can see the image looks much clearer already, right? You don't have a lot of background photons. I didn't really make a point of this, but if I go back, you can see the total number of photons, which you can read over here easily. Those in a single frame of peaking around 10 to the 3 photons to averaging 12 frames, you can see it goes all the way up to 10 to the 4 photons. I'm increasing my photons because I've scanned the same frame 12 times. Okay, and you can also see that my FLIM curve is much smoother, and we'll get back to that later. But basically, hopefully this gives you an idea of what a frame is. So you always have a choice of how many frames to average, how many frames to take, and how many frames to average. Usually we, well, that's not entirely true. For most imaging, unless you're imaging extremely fast, you'll always average the number of frames that you take, because we basically use frames just to increase our signal to noise, okay? If you want to do extremely fast imaging, that might not be the case. You might just use individual frames to analyze your data, because that will allow you to image very rapidly. But for most typical imaging that we do in the lab, frames are sort of a tool to increase our signal to noise, okay? All right, so if that's a frame, what's a slice? So a slice is basically a collection of frames, okay? So right now, this one slice is consists of 12 frames that an average together, okay? Now, we have slices in 2 different domains. One is Z stack. So when I talk about Z, what that means is, you know, in your 3-way coordinates, you have X, Y, and Z. So moving up and down in the slice. So a lot of times we like to take stacks of multiple slices because our dendrites, of course, aren't perfectly flat, right? And so if I focus up or focus down, I can see different features. So sometimes we might want to take a Z stack made up of multiple Z planes, okay? So if I have over here, I have slices. If I change this to 3 slices and I have this Z stack button clicked, it's going to basically take 3 sets of 12 frames at each Z position, okay? And I define the distance between the Z positions over here by step. You guys see my pointer, right?
Yeah.
Okay. So step, I have 1 micron. So basically it's going to take 12 frames, average together, move up a micron, take another 12 frames, and then move up another micron and take another 12. So it looks something like this. That's the first 12. There's the second 12. And here's the third 12, okay? And then it spits out a projection image. So what it's spitting out, you can actually see here, is a maximum projection. You can actually change this so that it doesn't project. And now it actually has the 3 slices separate that you can toggle through, okay? So usually when we're doing analysis, we're taking the Z-stack, we'll have the analysis done in the projection. So we usually keep this button here. But this sort of gives you an idea of what a slice is. You can see that, you know, you have 3 slices now in this image, okay, that you can actually toggle through, okay? So if you're doing something like structural plasticity on a secondary dendrite, you might use a Z-stack of slices, for example. The other way we use slices is not for a Z-stack, but for something called a time lapse, okay? So this is, the slice is the same thing. One slice is still going to be equal to a set of frames, okay? So in this case, again, a set of 12 frames that are averaged will be 1 slice. And then I can decide, instead of going, my slices being in Z, they're going to go in time, okay? So let's say I wanted to take 10 slices, okay? I want them separated by, let's say, 2 seconds, okay? So now it's going to take a set of frames every 2 seconds, okay, from 1 to 10, okay? So if I started this, you'd see that's 1 set of frames. Oh, wait, I'm still, oh yeah, okay. Hold on. Oh, it didn't change, okay. I was like, why didn't it take 10? Okay, so it just took 2 or 3. Let's try that again. Okay, so you can see it counting up the various slices here, and it's going to go all the way to 10, okay? And it's 2 seconds between each slice. You notice here it takes 1.5 seconds to do 1 set of frames, okay? So it's kind of slow. So I'm basically imaging almost continuously with a short time in between. We use time, we use slices in the time domain, usually if we're trying to image while we're trying to uncage. And the reason we do this is it allows a little bit of gap between images so that we can stick an uncaging event in between. And, you know, Molly and Anand will give you, you know, specific, you know, protocols for exactly what amount of frames and however many number of slices and how you're going to do it in order for each experiment. But you guys can also play around and see how it affects your data. Have an option to average slices. That's the same thing as averaging frames, except for it's going to be actually averaging, you know, multiple sets of frames or multiple slices. Okay, and again, you'll see your data comes out here as a series now of 10 different slices, 2 seconds per slice, and you can click through them. It's just like a time series, okay? So you could analyze, and I'll show you an example of a data set where I use slices, and I'll show you how I'll analyze something like that, okay? And then the last type of image we have is this loop image sequence, okay? So the thing with frames and slices is that the file is all a single imaging file, okay? So if I were to open this time series that we just did, it's just a single image, okay? With these slices embedded in it. Loops are actually different files for each image that you take, okay? So for a loop image sequence, it's going to read everything that you have up here, okay? So it's going to take all this into consideration, and all of that will be one image. Okay, so see how these are sort of embedded inside each other? You have frames as your first thing that you can manipulate, and then you can manipulate your frame sets into slices, and then you can manipulate your slice sets into images, okay? And so this gives you a tremendous amount of variability if you want to build complex imaging protocols. Typically, we don't do super complex things. We might use frames and slices and loops to do, for example, a time series with a Z-stack, okay? So I might use frames to increase my signal to noise, and then I might use slices to do a Z-stack so that I can see along a stretch of dendrite. And then let's say I want a Z-stack every 30 seconds. Okay, so here we're saying what our interval is between our images, okay? And you can also define the number here. So let's say I want 3, okay? When you set this up, one interesting thing is, whereas you start grabbing individual images from here, this grab button, if you want to start a loop series, you always have to hit loop here, okay? So now my loop series is going to be a Z stack. So again, it's doing its frames. You can see it counting up. And then it's moving in the Z, okay? Now it's in between. It did this whole set. Now it's in between these loops. So it's counting up to 30 seconds. Okay, you can see a lapse time here. This is really helpful. During this time, you can refocus if you want. When it gets to 30, it's going to start this whole image slice sequence again. Okay, whatever you have defined here. So now it's doing another one. Okay, and then it's counting up again. I'll let it do its last one so I can show you how it's saved a little bit differently. Okay. Okay, so now it's done. Okay, so what you can see is if I open these images, they're saved as separate images, separate files. Okay, you can actually see it. If I open the folder, you can see 3 different image files, image 13, image 14, and image 15. Within each of these files, you can see that I have Z stack of images. Right now it's shown as a projection, but I can undo that projection button and you can see the 3. And remember, each of those slices is made up of the 12 average frames. Okay? All right, so is that clarifying that a little bit? Questions? No? Okay. So these stacks
I have a question.
Yeah.
When you start doing the Z-stack imaging, do you set the stage first? Like, there is this option of reading the stage values. Do you set that before you start imaging, or?
There's been a bug in one of the softwares, which I think is mostly fixed now, that if you
you focus it and then it automatically detects it?
If you focus it, it should be automatically detected for the center. Okay. In the software, if you have a set of 1 slice, like if you're not intending to take a Z-stack, in the software version, I think it's been fixed now. If you don't hit set, it will move it. Like it thinks it's taking Z-stack. But generally, no, you don't have to. It will just read wherever the focus position is. That will be the center. But if you want to be extra sure, you can always hit center button. Okay. You'll notice you have this back to center, back to start, and stay. If for some reason you wanted to set your Z-stack from the lowest point instead of the center point, you can use this back to start. Stay I was using a lot when there was that bug where it was jumping around unintentionally. This just won't let your motor won't move if you keep it on stay. Okay. But usually you want to keep it as back to center. Okay, just so you guys know, this down here is another way of reading out your motor position. One thing that's really comes in handy is to try to see how deep you are if you're trying to determine if a spine might be too deep to uncage. Probably You guys might have heard that we tend to uncage on spines. I say 50 microns or above. Some people say 30 microns or above, because it's hard for the uncaging laser to penetrate really deep into the slice with full efficiency. But you're like, well, how can I possibly tell how deep I am in the slice? So it's not actually super easy, but what we do is we use this motor readout to try to determine how deep we are. I don't know if you guys have been shown that already. No? Okay. So what I do is I hit 0Z, okay? And you'll see that that gives me a Z value of 0, okay? And then if I hit focus, I try to focus up to the top of the slice. When I'm doing that, I change the contrast by just hitting this auto contrast button so that there's more noise. Okay, and so I just hit it again so that I can see the noise really well. Usually you can tell by the noise, watch me not be able to show you, when you're out of the slice. So, okay, so this is very subtle to see, and I don't even know how well it's transmitting over the computer. But right now, I'm out of the slice. I'm going to go down slowly, and you should see the noise increase. Okay, so going down. Okay, right there. Can you guys see that? The noise increased a little. Okay, and that's when the laser is starting to go into the slice. I can see the noise levels increase. It's not super accurate, but it's a quick way of sort of getting an idea. So now I can hit read, and now I can see that I was 20 microns before. Okay, so now if I focus down again to where I was, and I read this again, it's close to 0, right? Okay, so sometimes if you're not getting plasticity and you're like, my hand looks really healthy, why am I not getting plasticity? One thing to check is, are you too deep in the slice? Okay, if you're 80 microns deep, it's going to be really hard for the uncaging laser to uncage the glutamate efficiently. You'll also notice that your neurons tend to be much dimmer because, again, the fluorescence is hard to get out from that deep, and your excitation laser is a little bit hard to get in. Okay?
So the best way to see this is zeroing it at the spine that you're wanting to record from, then pulling back.
That's how I do it. If you prefer to zero it at the top of the slice and then go down to where you're focusing, it's the same thing.
Okay.
But just because usually I at my spine first when I'm wondering this, and I usually hit 0Z and then go up from there.
Okay.
Okay. Okay. You can manually move your motor. I almost never use these buttons. I almost always just use my, micro manipulator, but that's what these buttons are here. I assume you guys have been told about how the file naming works and setting your path, correct? And the importance of saving your sanity by always starting your experiment with a new base name and at number 1. It really makes a difference. If you can at all get used to doing that, it'll make your life easier when you analyze data. Okay, so now that we have that, basically, let's talk about some of the more interesting things, okay? So, Okay, so here, basically all you guys probably use on here is the zoom, okay? Zoom is actually really important.
Can you hear me?
Yes.
Okay, I have a quick question. How thick are the slices normally?
So we cut them at 350 microns, but as they grow over 2 to 3 weeks, they tend to spread out a lot. So we don't know exactly, but probably around like 200, something like that.
Okay. And you shouldn't be going down more than 80 microns, you're saying?
If you're uncaging on spines, I try to stay in the top
Okay.
50 or so microns.
Okay. Thank you.
You'll notice that if your slices are not very healthy, you often will get, your slices are not very healthy, but you still do get transfection. Often the neurons that are transfected are really deep. And that's because the neurons on top tend to be sort of dead, and so they don't express your construct. The deeper neurons tend to be more healthy, and you might get some transfected neurons there, but you'll find it's actually really hard to do experiments on those neurons because the signal will be dim and the spines will be deep. So you can try, but it can be difficult.
Thank you.
Yep. Okay, so Zoom. So why do I like Zoom so much? Zoom, of course, is, it's important for, defining what area you want to image. But one of the things that I think is somewhat underappreciated is if you have dim neurons, like I almost always do, and dim slices, the best way, if you can, to increase the brightness of your neurons is to zoom in. Okay, so by zooming in, you're getting, if you only are trying to uncage on one spine, depending on your experiment, you'll need to really image different distances, right? And, but I think sometimes we feel like we have to image a big segment of the dendrite, but really we're only interested in the spine. You can see this is pretty dim right here, but if I zoom in and I'm looking at this spine right here, for example, okay, look, you can see I have like very little signal here, right? Now I haven't taken my average frames yet, which of course makes a huge difference. Change this to one slice. And see I have focal drift. Okay, so let me change my contrast so I can see it better. Okay. All right, so if I'm looking here, I have, 10 to the 2, between 10 to the 2 and the 3, kind of noisy. But then if I zoom in, I've already zoomed in a lot. I'm at like 60, but let's say I zoom into like 80. Or even a little more. I'm going to get many more pixels out of my spine that I'm interested in. And now you can see I'm over 10 to the 3 for photons, and my curve is a lot cleaner. Now, of course, it'll depend on the type of experiment you do, and you may have to stick to a certain zoom, but I like to zoom in because it's a good way of increasing my signal. Now, when you zoom in, you could potentially be causing more damage because, right, you're exciting the spine more intensely. So that's one thing. If you have something that's prone to photo bleaching or something that's really sensitive, that's something to pay attention to. Okay, but don't be afraid of zooming in, especially if you have dim samples. If you're working with Kamui, you don't have to do this. Okay, so there are a lot of other things that this can do. You can basically image a certain region by changing the angle of the mirrors instead of the region that it's scanning, but you guys don't need to worry about that. Here, one nice thing is it shows you basically the size of your field of view. So at zoom 1, you've got this microns by this microns, and you can see zoomed in right now I have 5.7 microns with 5.7 microns. Okay, you can type in these things, or you can use these down arrows. You can also use this max button, which goes all the way up to 1. All right, scan. So again, probably these settings will be pretty set for most of the experiments that you do, but just so you have a quick idea what they mean, this basically defines your scanning properties. So right now I'm scanning at 128 pixels in each line. and 128 lines in each frame. And my line time, how long it's going to take to do that, is 1 millisecond. You can change these things. So if I changed it to 2 milliseconds, what do you guys think happened? Couple things. It'll take longer, right? Because now instead of 1 millisecond for each line, it'll take 2 milliseconds to go in each line, and then 128 lines down. Also, it got much brighter, right? That's because instead of exciting with 1 millisecond of time at each pixel, or each line, sorry, not each pixel. This is the time per pixel, 10.9 microseconds. I'm spending twice as long, so I'm able to collect twice as many photons pretty much. Okay, so if you're really desperate, you can scan slower and get more photons. But again, this is something you'd want to keep constant for your whole set of experiments probably. So this is something that you shouldn't just change experiment to experiment, but you know, you'd have to talk to your mentor about it, but just so you understand what it is. Another thing you can do is, instead of doing 128, if I do something like 32 by 32, it looks like this. Okay, so now you can actually see the individual pixels pretty easily. It's basically binning a bunch of pixels together. So instead of, you know, I'm losing resolution, I'm gaining, depending on how you set it up. If you keep the pixel time constant, you can gain speed, or you can gain photons per pixel if you keep it at the same speed but use less pixels. Okay, so that's just another trick. Typically we use 128 by 128. FLIM is a photon demanding technique, so we need a decent amount of photons. If you were to take a confocal image for like a paper in most other labs, you would be using something like 1024 by 1024, or even 2048 by 2048. Our images are relatively low resolution in the imaging world. But if you think about what we're actually imaging, like this spine is about, let's say, about 1 micron at the biggest across, right? Well, light microscopy has a resolution, you know, depending on the type of light microscopy, but something like 200 nanometers, okay? So we're imaging small structures. So having really, really high resolution, we're still not going to be able to resolve much within a single spine. So for us, having a lower resolution image isn't, for most things, isn't really crucial, okay? But yeah, unless someone tells you otherwise, you should be using 128 by 128. A lot of people use 2 milliseconds instead of 1 millisecond. I just use 1 millisecond. So whatever set on your microscope or what your mentor wants. Okay, enough on that. So SPC tab. This is basically everything you need, everything involving the conditions for your FLIM curve. So again, this is something you probably won't change unless you're having trouble, something changes on your microscope, something like that, okay? But just to give you a quick idea about sort of what some of these things are and what they mean, we can talk through a little bit. Okay, so let me get an image so we can get a nice one. Okay, so SPC, you have sync channel 1 and channel 2. Sync, what that basically means is it is the signal that's coming from your laser to turn on the stopwatch. Remember how we talked about FLIM being like a stopwatch? When the laser pulses, that's your start signal. And then when the photons are detected, that's your stop signal, okay? So the sync basically defines what the computer is considering the start signal, okay? So if you actually looked at the signal, I mean the electrical signal that's coming out of the sync cable in the laser, it's kind of messy looking. And you have to define what you're, in that electrical signal, what threshold you're going to use to define it as an actual. Okay. And if it's set that threshold wrong, so I did my minus 100. See if I can make it wrong. I don't know how to do this. I was just doing this. Oh. Okay, so that's not good. That made it wrong. Now I don't even get any photons. Hold on. Not what I intended to do. Oh, so now I have check laser setting. Did you guys see that? You might see this message. Let me go back and see if I can do it again. Okay, look. Now, I didn't show you this before, but this shows you what it's reading for the pulsing of the laser. Okay? Minus 100, it was reading 80 megahertz, which is what it should be. Now that I've changed the threshold of what it's counting as a pulse, now it's not counting any pulses. You can see it's 0 megahertz. And it says check laser setting, but the pulse rate is not good. Okay? Now, sometimes you might get something that's a little bit closer. And it might make your FLIM curve look kind of funny. Okay? So, and these are things you need to really change. This offset button basically shifts where your FLIM curve starts and ends. So, I think you guys had trouble with this one day. So, if I change mine to 2500, you'll see it's basically shifted at the start of it. And then this is not the start, and this over here is actually the tail. It should be over there. Let's see, 500. Okay. All right. So, shouldn't have to really deal with this too much. This resolution has to do with how many different points on your FLIM curve you have. Again, not really important unless you're like mapping like lifetime curve of a brand new fluorescent protein. I don't want to change the threshold. So this defines basically how much signal it's going to count photons. What amount of signal it's going to count as a photon in your image. Okay, so you'll see that I have readings down here of photon count. I'm scanning right now and I'm still counting this many photons per second. So that's kind of a high background maybe. You could argue that maybe I should change the threshold to something a little bit different to get these counts down. Once I focus, you can see it goes way up to 110 photons per second. Okay. So this is something, again, if you're having trouble, you might want to change this, but it's something you really want to touch once you're having trouble. Channel 1 and channel prefer the green channel and the red channel. Alright, and I think that's all you guys want to know about that. Again, this channel just allows you to tell the computer what data to collect. Usually we just collect data on both channels because it doesn't really have a negative other than using a flow memory or something, but that way you always have whatever data you need. Okay, any questions about that? Alright. Now calibration curves, I think probably you guys have already talked about that. You want to make sure you have a nice calibration curve, should be a minimum and a maximum of each lasers on. Measure your power every day before you start doing experiments. If your calibration curve ever looks a little wonky, this also should give you an error like this, error 3 decalibration. And yeah, you should ask for assistance if you have that. Could be a problem with laser, apocalypse, or something else. But okay, that's pretty much it. Again, this is a really nice readout to see that your system is working right. These are pulls always you want to have about 80 megahertz. If you're having trouble with your images, keep an eye on this total count to see what's going on. All right, so that's pretty much it. I'm going to come up here to show you a few things on tools. You guys already know about the paging control window. You guys are both on my paging, right? Okay, so we won't go through that. This image sequence control. This is a feature. Can you guys see that or no? Okay. This is a feature that allows you to change imaging parameters in the middle of an experiment automatically. So let's say I wanted to take these stackers, and then I wanted to do some really fast imaging just in one lane, and then I wanted to do really slow imaging for like 3 hours with this stack again. I could set out all those parameters using the image sequence. And load all those different settings, and then you just take run sequence, and then it will automatically change through the different parameters. So if that's something that your experiments need, we can go into more detail how to do that. Just know that it's working. If you need a little bit of troubleshooting, I haven't used it recently in the latest software. We're going to talk today about the drift control.
Also in the image sequence, once you create an image sequence, you can save that setting. So you don't have to really do that sequence again and again. You can also load that sequence.
Yes. And even without image sequencing, just saving your settings here. So the software always opens up whatever your last settings were, which is just super helpful. Also, like, if you were working on the computer with different sequences, you can save. Like, if let's say you set up, averaging these frames, and you wanted to do a complex, timeline series or something, you can save whatever your settings are here. Just doing these setting as, and then load scan parameters. Okay. That's really helpful because you don't have to always reset everything up. You can just save your settings. Okay, so the focal drift, I didn't try it a lot. I did try it some, but I was mostly working on primary density. It does seem to work. I don't know completely how well it works, like some secondary dynamics. But definitely it would get huge advantage if you didn't have to be sit in front of your computer and keep focusing all the time. It would make it much more efficient. So I definitely try that out. This is what we'll need that we talked about, that you'll need mitigating correction, you shouldn't need remote control, stage control, here it is. Okay. So, most of the other stuff, you shouldn't really need.
Maybe a real-time lot.
Yeah, so the real-time slot normally is open.
Send your those.
Okay. So, real-time plot basically plot with everything in real time, as the name suggests. On your focus, right? So I go out of focus, you'll see my intensity drop. I go focus, I back up. Okay, what's also is you can also plot in tau. Basically set it. Okay, so you can see this is just a GMP PKC delta. You can see it's hovering around 2.6. What's cool is that go out of focus, my intensity will drop a lot, but you can see that lifetime is basically independent of intensity. It's basically steady 2.6. It's getting noisier because now my FLIM curve is noisier and less accurate, less total, right? See how much noisier it's getting? But it's still basically staying around 2.6. I'm back in focus, it gets less noisy. But it's cool. Yeah, so if you're really focusing, sometimes there's this real time plot. I use it less than I use time course, which is super helpful you use during an experiment to sort of see what's going on. Okay. Let's go. Take these images. Let's go and take a look at this window here and your FLIM for below and these values. Okay. So let's say we just need to get a nice. Okay, so when you take an image, of course, a beautiful light center or no light center, depending on how many photons you have. How many photons do you need for an experiment? Well, in no set value, I will tell you my experience. Obviously, if you sign up in like 10 to the 4th range, your error will be like 1% or so. Very nice. This is a primary dendrite, so it's typically you're not imaging things right. And then to the 3rd range, I'd say is pretty acceptable. Good. You get an experiment with these low noise. If you start getting down where your peak is at like 10 to 2, it's sort of borderline. Your data might be quite noisy, and you'll probably have to average a lot of experiments together. And anything really below 10 to the second, I'd say is probably not worth doing. So try to see if you can find a way to get your photons up more to the ideally like the 10 to the 3rd range or so. Mid if you can. If you possibly think about turning up your laser, I don't normally do that too much, but if you do, that's a possibility. Averaging more frames, things like that. And if you consistently have low expression, you should think about what strategies you can use to sort of boost that photon count. Okay, this down here is a measure of error between it and your lifetime curve. Okay, so, all right, let's talk a little bit about this window in general. So, channel 1, channel 2, and then channel 1, 2. So this is, I have red things actually in the nucleus, but I'm at the nucleus right now, so I don't really have much red. These all bleed through. One thing that I see searching for are imaging techniques and more channels. If I had something in red, you could see the original, maybe it's signal there. So I sort of showed you how you can go through your images by using errors. Right, these buttons here, depending on if you're taking images or you have slices, you'll use this down here. Okay. Alright, so we do need to talk for a minute about contrast, because there are some values that will affect your actual data. Okay, so there's really only one value that will affect your actual data analysis, and that is if you're analyzing lifetime values. This lower value, the minimum value, will affect your data analysis. Okay, so. I'm not going to read data, but the way that lifetime analysis works is that any pixel that's colored black is not included in the analysis. Okay? And any pixel that has color in it is included in the analysis. This is only by lifetime. Okay? For intensity, that's not true. For lifetime values, that's true. So right now, you can think sort of on the edge of my neuron, I have these sort of reddish colors. Those are being included in my analysis. If I change this lower threshold, you can see that most of those pixels go away. You can imagine that will change my data somewhat. Okay, if I change it dramatically, really, I get less of a neuron, right? Say I change all the way up to you can start to see, all right, that's not dramatic enough. Hold on. You can start to see I'm actually losing a lot of photons here. Okay, so before my curve went all the way up, like where this red curve is, now it's dropped down. Okay, so what you want to do is you want to pick a value that's a faithful representation of your neuron, basically. And I know that this is a little subjective. The most important thing is, since we're always looking at lifetime change, typically, is that you just keep this value constant through your whole experiment, okay? So auto contrast is actually, I find, pretty good at getting this bottom value correct. I mean, when I say correct, what I mean is I would maybe agree that that's a faithful representation of the neuron. So if you want, you can use auto contrast, but you can also just sort of eyeball it and say, you know, I think this should be a little lower because I can kind of see that there's a spine here and I can't really see it over here. So, okay, now I'm starting to pick up that spine a little bit. Okay, so this is something that you'll just have to get used to. It is subjective and you might set it differently than I would set it, but as long as you're consistent throughout your whole image series, it's not a big deal. But you do need to know that this value will affect your data, so you should pay attention to it, okay? These other values, the top value of FLIM, for example, doesn't affect your data analysis. It will affect how it looks here, how the image looks here, but it will not affect your analysis. For intensity, however you set these values doesn't change your analysis at all, okay? It just changes how it's displayed. Okay, so you can set them however you want without worrying about if it's going to change your analysis. Okay. This right here, this lifetime color map, this is just for display. It allows you to sort of see, visualize what's going on, but actually to make images, like for a paper or for a lab meeting, you'll want to, this is really the only place to set a color map. This is basically showing your range. So everything as a lifetime value of 2.6 will be blue. Everything as a lifetime value of 2.1 will be red. Okay, so you can change this. And you'll see now my image is much more red if I change it to a smaller. And this is something that when you're analyzing specific data, you'll have to play around with to see what range allows you to show the change that your data is showing you, okay? This filter is another thing that we sometimes use for images. It sort of just smooths out the lifetime variability. So when you get making images, we'll help you a little bit. There are other tricks that we use, like you can actually do time binning. And this will basically average several lifetimes over like a very serious time period. So let's say you wanted to show a representative image of what the lifetime looks before manipulation, which is maybe like from -2 minutes to 0 minutes. And you could time in that. And then from 0 to 2 minutes, which is right after uncaging. And then from 10 to 20, something like that. Okay, and then once you can export images into TIFFs, and we can show you that when you're trying to do it. Okay, and we'll save your images according to the parameters you've set here. Okay, so how do you actually analyze data set? So maybe the best way to do this is for me to actually open up something. So okay, I'm going to open up the data set right here. So you can see that when it opened, it kind of did like a quick analysis. This is data that I took a little while ago. It has a sensor in it, and I can just go through quickly how I would analyze something like this. Okay, this is a data set using slice imaging. You can see it's one single file, and it has a time series of 50 images at 8 seconds per slice. Okay, so to flip through these images, I would click through here. Okay. Now, right now I have this calculate upon open checkbox checked. So every time I open an image, it's going to keep reanalyzing it. That can be advantageous or it can be disadvantageous. It just sort of depends on what you like. Okay, but now I need to look up here. First of all, I can see that this FLIM curve, I'm missing the top of it, right? So this is defined by this fit start and fit end value. So if we go all the way out, I think we have a range of about 1 to 60 or so, okay? You can see this is our full lifetime curve. But this beginning part is mostly dominated not by our actual signal, but just how long it takes the instrument to respond. We call that the instrument response curve. So we usually like to get rid of this whole tail. But we do want this peak. It's really important to have the peak because that's where most of our photons are, okay? Okay, so you can see as it gets to the end of the curve, I have less photons. This is pretty bright, so it's not too bad, but you might see it starts to get noisy here. So often we'll cut off the end of the curve, so it's less noisy. Okay, now I have a decent looking lifetime curve and I want to basically set my fitting parameters, okay? So again, this will depend on your sensor that you're using and your project that you're doing, but typically we set tau 1 and tau 2. So what is tau 1? Tau 1 is the lifetime of our donor fluorophore if it's not undergoing any FRET. For GFP, it's typically 2.59 or 2.6. However, if you attach GFP to a protein and you express it in neurons, it usually is slightly different because the environment of it is slightly different. And this is something actually that for a new sensor we typically would measure by just expressing the donor part of our sensor and just expressing it in either cell lines or neurons, or and taking a bunch of images and doing a single exponential fit. Okay, so single exponential fit basically means there would be no FRET going on. You wouldn't have any acceptor, so there couldn't be any FRET going on, and you'd see what the best value of tau 1 is. Okay, for my particular sensor, I've measured that value to be 2.65. If this hasn't been measured in sensor, you know, ask your mentor, but an estimate of around 2.6 is probably, if you're using GFP, is probably a good estimate. Okay, so we typically fit that value. Tau 2 is a little bit more complicated. What it is it's the value of, it's the fluorescence lifetime of the donor if it's undergoing 100% FRET. That's the definition of it. What it is in practice is a little bit hard to measure. We normally estimate it, again, in a situation where your sensor is undergoing high amounts of FRET and using a double exponential fit and estimating what tau 2 is. Once we have a good estimate of it, we usually fix it. The good thing is that the actual number that you put in here for tau 2 really doesn't affect your data hardly at all. Like if I put in 1 or 1.3, it really changes almost nothing in the data. I've actually tried this, so I feel confident in saying that. Okay, so once we have those two values fixed, for most sensors, you'll fix those values throughout the whole experiment, okay? So you fit those two values, and then what I'm gonna do is I'm gonna fit my lifetime curve, okay? By clicking this fit button. Okay, and when you do that, it populates a good fit for these other two values, tau G and tau 0, okay? Tau 0 is basically a measure of what we call the offset, or you can think of it as the system response function. Tau G is also a measure of the width of the Gaussian of this system response function. I don't have, honestly, a super clear understanding of how, of these exact values, because if that is totally true, the way, and maybe Long, is Long still here? Yeah, Long is still here. If he wants to chime in, if he's still listening, he can. But to me, the value should be fixed across all experiments. It's a measure of your system response, but they're not. They're dependent on your system. And each experiment you do, we fit them again. So I don't totally understand that. They're calculated though from the fit. So they're not like a measurement of your system response function. So it's a mathematical calculation. And I can show you, actually, it's, you know, in all of our papers, it's published exactly how these values are calculated mathematically. But basically, you should think of these as measurements of system response function, okay? So we're gonna fix all those values, okay? And when you do that, you get a couple measurements that pop up here. One is population 1. This is the amount of your sensor that is not undergoing fret. This is population 2. This is the calculated amount of your sensor that is undergoing fret. And then a mean tau, okay? So this is basically not based on fitting. This is basically the average of the lifetime in each of these pixels, okay? And you'll see that right now, this is calculating everything for this entire region. But if I were to draw an ROI here, then I could calculate these values for just this ROI, okay? Again, so you might, in a typical experiment, want to calculate multiple values for different ROIs. So maybe like, the spine and the dendrite, okay? Let me actually remove these because the spine that I actually stimulated is this one. So let me make this ROI 1, and then I'll make a dendrite ROI. Okay, so now that I've fit all these values and I'm ready to basically go through and calculate these values, okay? So what I'm gonna do is I'm basically just gonna move through each of these experiments, okay? And I can adjust my ROIs as needed, okay? And just gonna go ahead and calculate these values. You can see this is starting to get redder in the dendrite, in the spine. I changed the loot, you can see it maybe a little better. Now my spine is starting to grow. Okay, and different people might do analysis slightly differently. So just check with your mentor to make sure that the way you're doing it is the way they want it done. If you move your ROI, I haven't been showing you this because it's not really doing real analysis. If you move your ROI, you need to re-hit this calculate current page button, okay? And then as you open your next image, it's automatically analyzing because I have this calculate upon open, okay? Okay, and you can get plotting ROI 1 and ROI 2 down here. So now this is going to focus. So I'm going to move these and I'm going to hit calculate current page. Okay, you can zoom in on this so you can see what's going on a little better. To zoom out, you double click. This automatically scales, so until all these things have a value, it will stay zoomed out like this. But okay, so this is what my experiment looks like. ROI 1 is the spine, ROI 2 is the dendrite. Okay, you can see here I started on KG and I had this drop in fluorescence lifetime. First in the spine and then in the dendrite, and then it sort of came back pretty much to baseline after that, okay? You can see this is in fluorescence lifetime. If I switch to fraction 2, that's another way of saying basically population 2, okay? The percent that's undergoing fret. No right way of which one to look at, it just depends on what you want to look at, okay? Intensity sum. This is what we typically use for looking at structural plasticity. You can see this is my spine down here. So if I were to zoom in, you can see with uncaging, I got spine increase, and then it returned not quite to baseline, about 1.3 to, so sort of mild plasticity, but still some plasticity that's sustained, okay? Okay, so what makes, well, before we get into that, when you have it analyzed, this automatically gets saved in an Excel sheet, okay? So if you want to see that Excel sheet, you can go to view and, hold on. And this is the Excel sheet where it got saved, okay? What you have here is a measure of time, file number, time in seconds. Okay, then you have your data. So lifetime for ROI1 and ROI2. We have two channels. Channel 1, this is what we're actually interested in. Channel 2, I didn't really have any signal, so this is just like data I can ignore. I have lifetime and lifetime fit. What's the difference between those two values? So lifetime fit is dependent on the fitting of your curve, okay? Lifetime is independent of your fitting, or mostly independent of your fitting. Usually we use lifetime fit. If you have a good fit, these values should be fairly similar, okay? You can see they're not identical by any means in my case. But if we were to graph the two of them, what you would see is that basically the graphs would be almost identical, okay, even though the values aren't. So often when I'm doing analysis, I do a quick graph here just to sort of see what happened. You know, it should look pretty much the same as my other graph from my real-time plot, okay? So this is basically where all your data will go, and it gets saved it gets saved in this file folder under analysis. So this is your imaging file folder. You'll have all your images here, and then under analysis, you'll have your Excel sheets, okay, that you just did. And one thing you can do that's really nice is that once you've done some data, you can, okay. So when you hit this Python script, it opens up this window called average time courses. This is what you can use to normalize your data to baseline and also average a bunch of data together. So what you do is you define your baseline. So your images or frames before uncaging. And then it will plot a little graph for you. I sort of usually ignore this graph, so I don't bother changing all these, but you can. And then you tell it where to find the data that you want to average together. So let's try this. I don't actually know what data this is, but I'm averaging. Calculate. Okay, let me share that. Hold on. Let me share. Okay, so when I hit that calculate button, it basically made this graph here. This looks awful. That's because this data I'm doing some weird things in, like uncaging and then going back to the nucleus and imaging. But what this does, more importantly than make these graphs, is if you go back to your files, it makes this average here for you. Okay, it makes a new Excel sheet. And if you open that, what's really nice, okay, you can see that new Excel sheet, right? What's really nice is now you have all of your data normalized to your baseline. So what do I mean by normalized to baseline? So typically when we're talking about intensity, we want intensity values to start around 1, hover around 1, and lifetime values we like to talk about as change of lifetime. So we want them to hover around 0. So our initial data, sorry, I'm trying to show multiple things at once. So, you know, our initial data, our actual lifetime values are around 2.3, right, for our baseline before we uncaged. But what we want to do is we want to average the values across this time before we uncaged, and then we want to subtract that average out from each of these values. When new people enter in the lab, I always make them do this manually first so that they know what they're doing. But then once they've done it, then they can use this nice automated way of doing it, okay? And when you normalize to this baseline, then you're gonna have values that start that basically start around 0. So let's see, lifetime fit. So I'm looking here at lifetime fit values for channel 1, ROI1. Ignore this first one because it's messed up, but you can see that they're hovering kind of around 0. You can see this data's not super great baseline, but you can see it's hovering around 0, right? It goes back to 0. Okay. And so it has its normalized values for each of the files that were in the location that you told it to average, and then it has a mean value and a standard error of the mean. So what I would do is I would copy these files out and put them into a graphing software like Prism, something like that. Okay, so before, I just wanted to talk a little bit about what do you need to be careful of to have good quality data? So one of the most important things is having a very nice baseline. So in order to tell if your experiment is working, you don't want to have a baseline that's drifting. Okay, if your baseline is heading down like this, when you start your manipulation, it's really hard to know if your manipulation had any real effect on your baseline. So one of the most important things, and things that's usually hard for people who are starting out imaging, is getting a pretty flat baseline. So you can see that my baseline is pretty noisy here. It's bouncing up and down, but it doesn't have any real trajectory, right? It's not like heading all the way up or heading all the way down. Okay, so you want something that's fairly stable. You can see when I have more photons in the dendrite, I get a much, in this blue curve here, it's much less noisy, right? It's not bouncing up and down as much, and that's because I have more photons than I'm averaging in. So what can make a drifty baseline? It could be just that your neuron's not settled. When you put it in ACSF, it needs time to settle. It could also be photo bleaching, something like that. So it's important that you have a nice flat baseline. Wow, I just saw the time. Yeah, I mean, you end up not using a ton of it. So, and the more you play around, though, you shouldn't feel worried about touching any of the buttons. And it's sort of fun and educational to play around. So I got pretty much most of it. I think, yeah, just like what I was saying right now is just, having, a good number of photons, having a stable baseline, and having a baseline that's long enough to know that it's stable. So typically you do like 1/3 of your total experiment time or so. Again, your mentor will be able to give you really specifics on for your experiments what you're doing, but yeah, I think you guys got a pretty good idea. of everything. And once you guys have actual data, you know, the first time you do analysis, it's good to sort of go through with your mentor or with me or someone just to make sure that you're doing it correctly. But it's pretty easy. We didn't talk about things like background subtraction, but for most things, that's not really important for what we're doing. So